Hello, I'm Hannah Donnert with the Collaborative on Health and the Environment. Chanjoy is bringing you the latest environmental health science through our partnership calls, webinars, science serves, publications, and social media. I would like to welcome everyone to Che's third webinar in our six-part series on cancer and the environment, science and opportunities for prevention. Today's webinar is titled EDCs, Glyphosate, Arsenic, and Cancer Risks. I will serve as your host and moderator. We will leave time following the presentations for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentations, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone in the webinar right now is muted with the exception of our speakers. This webinar is scheduled to last for 70 minutes and is being recorded for our call and webinar archive. Before I introduce our speakers, I would like to update you on our Cancer and the Environment webinar series. This is a six-part series that was launched in February. In March, we held our second webinar in the series on women's occupational chemical exposures, nitrates in drinking water, and particulate matter composition and air pollution. In addition to this webinar we are presenting today, we have 3 additional webinars scheduled for the following months. On May 20th, we have a webinar on chemical mixtures in cancer, followed by a webinar on June 3rd, with speakers presenting research on environmental contributors to childhood cancers. To conclude this webinar series, we will present our final webinar with a look at disparities in cancer risks. This will take place on July 22nd. You can find more details to all of our webinars at healthandenvironment.org. With that, I'm very excited to introduce our 3 speakers here with us today, who have been involved in pivotal research on exposures to BPA, glyphosate, arsenic, and cancer risks. Dr. Gail Prins is a Michael Reese Professor of Urology and Physiology at the University of Illinois at Chicago and the co-director of the Chicago Center for Health and the Environment and UIC School of Public Health. She is the director of the University Andrology Laboratory at UIC and has been active in translational and clinical male reproductive research and activities for over 35 years. Her basic research focuses on estrogen actions in the prostate gland, including the influence of early life exposure to endocrine disrupting chemicals and toxins on adult prostate cancer risk. Her work established that early life exposure to natural estrogens, or EDCs, including bisphenol A and arsenic, permanently reprogram the prostate and increase its susceptibility to cancer with aging. Next, we will hear from Dr. Leanne Shepard, Professor of Biostatistics and Environmental and Occupational Health Sciences at the University of Washington. She served on EPA's FIFRA Scientific Advisory Panel for the evaluation of the carcinogenic potential of glyphosate. This led her to publish 2 review papers on glyphosate and a letter to the editor discussing the exposure estimations approach in the agricultural health study. We will close our webinar with a presentation by Dr. Tae-Hyun Roh, Assistant Professor in the Department of Epidemiology and Biostatistics at Texas A&M University. He conducts environmental epidemiology research on the long-term health effects of arsenic exposure from drinking water. He has a broad background in environmental health with specific training and expertise in toxicology and epidemiology. And with that, I would like to pass it off to our first speaker, Dr. Gail Prins. Feel free to take it away.
All right. Thank you everyone for tuning in. I want to talk today about the work that was introduced already on how endocrine disrupting chemicals can influence cancer susceptibility in adults. I'll show evidence that this may be in part mediated through the stem cell reprogramming. And I'm going to use the prostate gland as the organ that I show the evidence for, but I just want to point out that stem cell reprogramming by endocrine disrupting chemicals is now shown to occur in other hormone sensitive organs as well. So it may have broad applications. Oh dear. How do I? Oh, there we go. Okay. So The work that I do is embedded within a paradigm on developmental origins of adult disease. And in that paradigm states that early life events during fetal life or shortly after birth may influence the risk of disease later on in life. And this takes place on a number of different levels. With regards to the prostate gland, work from my laboratory and other people's laboratory for the past few decades has shown that the risk of prostate cancer is likely influenced by early life exposure to estrogens. And so there's a lot of work showing that this is true for maternal estrogen exposures. If the mother exposures are particularly high during pregnancy, the sons have an increased risk of developing prostate cancer as they age. Using rodent models, we've also been able to show that pharmaceutical exposures such as diethylstilbestrol or the birth control pill ethinyl estradiol can likewise influence prostate disease with aging. But pertinent to today's talk, endocrine disrupting chemicals are now being examined to see whether they might similarly influence prostate cancer risk. And so just to put everybody on the same page who might not be familiar with what are endocrine disrupting chemicals, they're a broad base of a number of different chemicals that are out there, synthetically produced. Some of them are herbicides, there's pesticides, fungicides, insecticides, and then there's a lot of industrial chemicals. There's thousands of them, if not tens of thousands of these chemicals that are produced that have properties that can interfere with the hormone system. Ones highlighted here in pink have been shown very clearly to bind to the estrogen receptor and activate the estrogen receptor inappropriately and have adverse effects. Others in bright blue have been shown to bind to the androgen receptor, the male receptor for testosterone, and actually block androgen effects. There's other endocrine disruptors that can influence steroid hormone synthesis. The one here in red, the phthalates, have been shown to interfere with testosterone and estrogen synthesis. And then there's other chemicals that can bind to thyroid receptor and actually inhibit its action. There's a number of other pathways as well. But just broadly speaking, these are chemicals that interfere with hormone action. And so the one that we study in my laboratory that I'm going to show you today is bisphenol A. And bisphenol A is not necessarily the most potent endocrine disrupting chemical out there, but it's so prevalent in our day-to-day life. It's one of the highest volume chemicals that's produced worldwide. And here I'm showing you the structure of a bisphenol A monomer. It's got these phenolic rings, and these phenolic rings are able to bind to the steroid receptors and activate them or inhibit them in inappropriate manners. There's thousands of products that are made with bisphenol A as a base, usually long polymers of them, but they can break down on repeated wear and tear or heating. Epoxy can linings, polycarbonate plastics, thermal paper receipts, many, many others. And we know that most humans are being chronically exposed to BPA because it's cleared very rapidly from the body within usually 6 to 24 hours. Yet when the CDC tested humans, most of them had it present in their urine, indicating that they were being chronically exposed. And likewise, the human fetus and neonate has also been shown to be chronically exposed. So in my laboratory, to address this, we used the model that I've been using for quite some time to look at estrogenic exposures, particularly pharmaceutical estrogens. And we gave bisphenol A either by a subcutaneous depot injection or by oral feeding to newborn rats at relatively low doses that humans might be exposed to on days 1, 3, and 5 of life. And in the rat, that's when the rat prostate develops. And we allowed these animals to age and looked at them when they aged. And when we looked at their prostate glands, we found pretty much nothing. They looked pretty normal and didn't have any observable defects by histology. It turns out that there were actually a lot of molecular changes that were taking place that were allowing the gland to be reprogrammed. So what we did next is we exposed these animals to estrogen as adults. And why did we do this? We mimic what's going on in the aging male. We know that with aging, males' testosterone levels decline and their relative level of free estrogen levels increase. And these estrogen levels have been shown to drive prostate disease. So we gave the animals adult estrogen exposure if they'd been exposed to BPA or vehicle. And what we found was that if they were exposed to BPA, there was a significant increase in prostate cancer susceptibility. So I'll just show you a little bit of data showing that. Here is data from our recently published Clarity study. This is a large study that was done with the FDA, consortia linking academic and regulatory insight on BPA toxicity. And here the FDA treated all the animals and at their headquarters, and then sent the tissues to a number of different investigators to look at the effects. And the tissues were all coded so that we could not be biased by their treatment. One study that they did was they gave BPA at different doses, starting on embryonic day 6, and they discontinued it just as the animals were weaning. And then in adulthood, they gave this higher level of estrogen and constant physiologic level of testosterone. And what happened when we looked at them at 1 year, we looked at the lateral prostate lobe, which is the lobe that resembles the human gland. And we didn't find a difference in incidence of prostate neoplasia in that lobe, but there was a significant increase in the severity of that neoplasia, going from low grade, which might be clinically insignificant, to high grade. And if they'd seen ethinyl estradiol as a positive control or the lowest dose of BPA, there was a significant increase in severity. This was maintained throughout all the different doses, but a little bit lower. We consistently saw the highest effect with the lowest dose. But more importantly, when we looked at the actual cancer that developed in the dorsolateral ducts, we found similarly that there was not a difference in incidence, but the multiplicity, the number of cancers that showed up, was markedly increased, very significantly, over 5-fold, if they'd been exposed to BPA early in life. Well, how might this take place, we ask? Well, here's a cross-section of a prostate, normal prostate in a human. And it's basically a very simple columnar epithelial gland of luminal epithelial cells that secrete prostatic fluid, lined by a layer of basal cells and sitting in within a fibromuscular stroma. And here's a schematic showing that. Well, we know from our studies that during prostate development and throughout life, there's this really small population of stem cells that are very long-lived, and they proliferate and maintain the structure of this epithelium throughout life. And this is how the stem cells get to a differentiated luminal and basal cells. We have a stem cell residing there within a niche. Every once in a while, it goes through a division. This division can either be symmetric self-renewal to give rise to 2 daughter stem cells, but the majority of the time it goes through asymmetric division, giving rise to 1 stem cell and then a daughter bipotent progenitor cell that then starts dividing and lineage committing either into a basal progenitor, which forms the basal, or a luminal progenitor, which forms the luminal. So we ask the question, might endocrine disrupting chemicals or BPA influence these long-lived cells? Could it be reprogramming them and setting the stage for disease later in life? So in the Clarity study, we've done this a number of ways, but I'll show you the data from the Clarity study, where they gave the lower doses of BPA, 2.5 up to 250 micrograms per kg per day, or the positive control ethinyl estradiol. And they did that every day up until 6 months of age. And then they shipped the dorsolateral prostate to us overnight on ice. And we immediately dissociated that tissue, took the epithelial cells and put them into 3-dimensional culture. And the majority of the cells are not able to survive, but the stem cells here in red are able to not only survive, but they start rapidly proliferating and forming those daughter progenitor cells. And after 1 week, we see these prostospheres, we call them. We disperse them, put them back into culture, and do that again after the 3rd generation, all the while exposed to no hormones. And then we looked at the properties in the 3rd generation of spheroids. And once again, we were blinded to the treatment groups, and when the data were decoded, this is what we found. They've been exposed that whole while to ethinyl estradiol, or the lowest dose of BPA. There was a doubling of the stem cell numbers within the organ. There was a doubling of the prostosphere numbers, which are representative of the number of stem cells within that organ. We then looked at their size, and with the greatest effect at the lowest dose of BPA. The 2 higher doses, we didn't have a large N number, and there was a lot of variability. But when we looked at their overall size, which is indicative of how fast those daughter progenitor cells can divide, we found that, once again, the positive control showed a major effect, as well as the medium dose of BPA. So we then took those spheroids and did expression analysis for a number of genes to identify the basal progenitor pattern and the luminal progenitor pattern. And what we found was that ethinyl estradiol and the 2 higher doses of BPA significantly increased the basal progenitor pattern at the expense of losing the luminal progenitor's pattern. So in other words, BPA and ethinyl estradiol were shifting lineage commitment in the prostate gland. So to just summarize this portion, we give BPA chronically. At the lowest dose, it increases the number of stem cells, and at a bit higher doses, it starts increasing the number of basal progenitors at the expense of luminal. So how might these stem cell changes influence prostate cancer susceptibility? We're not really sure. That we have some evidence, but I'll just point out that cancer risk has been shown out of studies from Johns Hopkins to be highly correlated to the number of times a normal stem cell divides in a tissue, including in the prostate gland. And we also know that tumor-initiating cells for human prostate cancer are largely localized to the basal cell population. So we're proposing that this chronic in vivo low-dose exposure, by increasing prostate stem cell numbers and altering lineage commitment, is underpinning an increased carcinogenic risk with aging. So might this apply to the human prostate? I would say yes, and I'll quickly show you why. So when we study the human prostate, we take prostate epithelial tissue from young organ donors, so they're disease-free. And we put those prostate epithelial cells into primary culture and then very quickly into this spheroid growing system that I've already described. And we're able to, after 7 days, get these nice stem-like and progenitor prostate spheroids. And we published on this extensively now, confirming that that's in fact what they are. And when we looked at them initially, we found that these spheroid cells had very high expression of estrogen receptor alpha and beta, which may explain why they're particularly sensitive to estradiol and BPA. And so when we give estradiol exposure, it increases the number of prostospheres and their size from the human. And likewise, if we expose them to BPA, we see a significant increase as the dose increases in the number of spheroids and their overall size at levels that are relevant to human exposure. And when we marked with BRDU specifically for the stem cell population, we see that BPA is really increasing those numbers at the lowest dose. So could this influence prostate cancer susceptibility in the human prostate epithelium? That's hard to do because men don't get prostate cancer until later in life. So what we did is we took those spheroids, dispersed them to single cells, and mixed them with embryonic urogenital sinus mesenchyme. And then we grafted those cells under the kidney capsule of nude mice. And after 1 month, we get this nice little structure growing on them. And in fact, it's a mini prostate gland. And if we stain for PSA, which is present in the human but not in the rat, we see PSA positivity. So we know we have like a humanized prostate structure growing in those mice. We then take those mice and we treat them with testosterone plus estrogen pellets over the course of a number of months. And what that elevated estrogen is capable of doing is increasing hyperplasia, prostate neoplasia, and actually prostate cancer. We start seeing that between 2 to 4 months at a relatively low incidence. So once we developed this model, what we did next was we gave these animals BPA during that 2-week window when the gland was forming. And this dose was equivalent to what we see in humans. We also gave another group BPA in vitro to the stem cells directly as well as in vivo. And then we did the T+E starting at 1 month and did blind pathologic analysis thereafter. And just focus here on the pink bar. If they'd been given oil during the first 2 weeks of grafting, there was about a 13% incidence of cancerous lesions. But if they saw the doses of BPA, the cancerous lesions went up significantly. And if the stem cells plus the in vivo exposure, it went up even higher to half of the animals now showing these cancerous lesions. So I'll close by just suggesting that we have a normal stem cell in the prostate gland. It's long-lived, it maintains the gland throughout life. If we see a first hit exposure to an endocrine disrupting chemical, that can reprogram these cells, and we have all the molecular data showing that in fact they are epigenetically reprogrammed. And this leads to modified gland. It may not be sufficient for cancer in and of itself, but if we give it a second hit, such as elevated estrogen levels with aging as occurs in men, if it's been reprogrammed by this first hit, it now starts developing preneoplastic lesions, which then can progress to prostate cancer at a higher incidence rate. So with that, I'll close my talk and hand it over to Hannah.
Great. Thank you so much, Dr. Prins, for your presentation and amazing work you're doing. We're going to have Dr. Leanne Shepard pull up her slides here. While she's doing that, I just want to remind everyone, you can type your questions into the Q&A feature available on the menu bar at the top of your window. At the end of the webinar and presentations, we will get to some of those questions, and we'll pass this on now to Dr. Shepard to start her presentation.
Yes, thank you for inviting me. And today I'm going to talk to you about glyphosate and the updated meta-analysis for non-Hodgkin lymphoma that we did. So many of you may well know that glyphosate is the most widely used herbicide in the world. It's sold commercially as Roundup. And the technical form of glyphosate is combined with inert ingredients to form glyphosate-based herbicides. And it's important to recognize that what's sold as Roundup around the world varies because of these additives, and they vary by location, even though they're labeled the same. Another feature about Roundup is that it's used as a desiccant prior to harvest that we've been calling green burn down. Glyphosate's incredibly controversial. In 2015, International Agency for Cancer Research declared that it was probably carcinogenic to humans, group 2A. The European Food Safety Association said it was unlikely to pose a carcinogenic hazard, and the Environmental Protection Agency concluded it's not likely to be carcinogenic to humans at doses relevant for human health risk assessment. So why did I work on glyphosate? Well, I served on the EPA FIFRA panel to evaluate the carcinogenic potential of glyphosate. There was some controversy around that because there was some industry complaints about one of the panelists, and I substituted. So I had a pretty short time frame in which to absorb an awful lot of material, in which I didn't really know much at all about glyphosate. However, the experience made me pretty concerned with what I saw that EPA was doing and how it was using the evidence. And so I joined 2 other panelists to address these concerns in the literature. I also had a personal reason for joining the panel because I have an organic garden, and I had thought that glyphosate was safe to use in restricted settings, and I wanted to know whether that was true or not. So the publications that I have a letter to the editor about the agricultural health study, a review of glyphosate exposure studies, and the updated meta-analysis I'm going to talk to you about today. So in that meta-analysis, we asked whether or not glyphosate-based herbicides are associated with an increased risk of non-Hodgkin lymphoma. We combined the evidence from 6 published epidemiologic studies, the 1 large cohort study, which is the agricultural health study, and 5 case control studies. And what was different is that we focused on the most highly exposed group in each study. And that was to get a better answer to the question about whether glyphosate-based herbicides are carcinogenic to humans. And at the time, there was a recently published update to the agricultural health study, which included a lot more follow-up, which we incorporated, and previous meta-analyses had not used that study. So I wanted to talk a little bit more about our exposure group, because that was one of the important features of our meta-analysis. And the reason that we chose the highest exposure groups is the relationship's more likely to be detected with higher exposures. We're less concerned about residual confounding, and it prevents the dilution of exposure groups and ensures a higher contrast. And so over on the right shows you the ordering of the highest exposure category, where we use the highest cumulative exposure and the longest lag because of our biologic understanding of cancer development, followed by other less strong characterizations. And there were several studies in that group of 6 that we could only use ever exposure, but we didn't want to drop them out. So we found that among the most highly exposed workers had a 41% increased relative risk, with a 95% confidence interval of 13 to 75%. And our results were robust to a number of sensitivity analyses we completed. I note that our results are consistent with, although higher as you would expect given the way we selected the exposure groups, with other previously published meta-analyses, including IARC and a paper by Chang and Delzell. And this gives you the meta-analysis forest plot of the results. So here's our primary result and all the studies that were included, and gives you a sense of the weight of the studies. And then also the parallel result using the older published version of the agricultural health study. So there's a number of strengths and limitations. We used the updated agricultural health study and we focused on the highest exposure group. However, there were only 6 studies. There's a potential in any of these studies for some publication bias. I think the numeric estimate, while it's a useful guideline, has some limitations because there were some main differences in the studies. For instance, their choice of reference groups that would suggest that it's kind of an apples and oranges summary of a bunch of risk estimates. And so I wouldn't put as much weight on the 41%. And the other feature that I'll come to a little bit is there's been a huge change with this adoption of the green burn down practices that these studies didn't incorporate in terms of exposure. So this supports IARC's conclusion that glyphosate is probably carcinogenic. I note that it applies to the most highly exposed workers, and so we don't know how that translates to the general public. However, as my work in air pollution also shows, with a ubiquitous exposure, even a small increase in risk means many more cases in the general population. And there are no studies of glyphosate-based herbicides impact on the public. They're a lot harder to do, but it doesn't mean that there's no risk because the studies haven't been done. So another point I want to make is there's a whole lot more to learn because the studies, all of them, the exposure started in 2005, and there's been, or earlier than that, some of them were much earlier, and there's been an explosion of use in recent years of glyphosate. It's actually the most intensively used herbicide in the world. And it's actually not only that, but also the most intensively used agricultural chemical of any kind. So this timeline is in our paper, and this just makes the point I was making earlier about the timing of the epidemiologic evidence, which is on the right-hand side, versus the usage of glyphosate, which is on the left-hand side. And the exposure assessment in all of the studies ended by 2005, and some of them were much, much earlier. Some of the case control studies were much, much earlier. And so we don't really know how this trend in glyphosate use worldwide has affected incidence because those studies haven't been done. So I also wanted to talk about, put this in context. So there's supporting evidence from animal studies, specifically malignant lymphoma and several other cancer endpoints, which I will show you in a minute, pooled analyses. But there are a number of limitations with those. A number of them weren't really followed long enough in terms of cancer incidence. And they also focused on pure glyphosate, and there's evidence to show that glyphosate-based herbicides are more toxic. Some of the mechanisms that have been hypothesized are immunosuppression, inflammation, endocrine disruption, genetic alterations, and oxidative stress. So here is a table from the recently published Portier paper of the animal study evidence with indication for the ones he evaluated: clear evidence, some evidence, no evidence. And 8 tumors show clear evidence in at least one species, strain, and sex combination, and an additional 3 show some evidence. So what's the broader context of this widespread use of herbicide? Well, herbicide-resistant crops are 85% of the world's genetically modified crop acreage, and the vast majority of these crops are Roundup ready. There's been a huge development of superweeds, which are herbicide-resistant weeds, and almost 50% of farmers surveyed reported glyphosate-resistant weeds on their farm. And there have been 38 weed species that evolved resistance to glyphosate that have been distributed across the world, and a number of different crop types. And this is probably the greatest threat to sustain weed control in the world. There's also been reduced populations and diversity, evidence of it affecting milkweed and monarchs, insects, and of course there was that horrifying report of the reduction of the bird population, which could also be impacted by the widespread use of herbicide. And there may also be impacts on soil health. Another aspect to consider is other herbicides, for instance, dicamba and 2,4-D are more acutely toxic, and their application requirements are stricter, which means there's more protections for workers and off-target effects. Glyphosate, in contrast, was considered safe for decades, and so there have been lower worker protection standards, and the tolerances or the residues allowed in foods over time have been increased. And as I mentioned earlier, it's the single most agricultural chemical in the world. So there have been some recently approved herbicide-resistant crops for glyphosate and other herbicides, and this it suggests a new era of pesticide pollution with no anticipated reduction in glyphosate usage, and the other pesticides having more off-target effects, drift, and due to their volatility. And then the weeds are developing a stacked resistance. So my conclusions, glyphosate is likely to be carcinogenic to humans. There's positive evidence in multiple sexes, species, strains, and tumor sites. This is strengthened by other lines of evidence in human studies as in our meta-analysis and some genotoxicity evidence, which I didn't talk about. I think it's important that there be a new paradigm for scientific review of studies that are funded by registrants and are used as a basis for policy, since there's a vested interest in certain scientific results. And in general, it's important to reduce pesticide usage and population exposure. And as I mentioned from my air pollution research, ubiquitous exposure with small adverse effects can harm millions of people. So this is a set of references that I will just leave up for a second and you can look at on your own time. And I want to thank you.
Great. Thank you so much, Dr. Shepard, for this presentation. Offers a lot of great resources that will be up on our website after this, and they're actually there now if you'd like to take a look. All the slides will be there. I'm going to pass this on to Dr. Rowe. If you want to pull up your slides, and I just want to remind everyone, I'm getting some great questions in right now. Feel free to put your questions in the Q&A feature on Zoom, and we'll get to them at the end of the webinar. All right, Dr. Rowe, you all set? I'm going to take you off mute, Dr. Rowe. There you are.
Okay. Good afternoon. Thank you for having me today. And I'm very excited to be able to share my research on the health effects of arsenic exposure from drinking water with you today. So my name is Taehyun Rowe, and I'm an assistant professor at Texas A&M University School of Public Health. So first, I'll give you a brief introduction of arsenic and its health effects. Then I'm going to talk about two questions I explored while conducting this research. Okay, so let's talk about arsenic. So as you remember, the title of today's talk is Arsenic, an Invisible Poison in Drinking Water. So you may have heard about arsenic poisoning cases from novels or movie, and even in history. So arsenic was used a lot to kill kings because when it is mixed in the water, there is no smell, no color, or no taste. So that's why its nickname was the poison of kings. Arsenic is a naturally occurring element existing in high levels within the Earth's crust. So depending on the geochemical conditions, arsenic is released and leached into the groundwater. So as you can see here in this map, many countries have a problem with arsenic contamination in their groundwater. Some Asian countries, European countries, even in the US. So some South American countries also have a problem with arsenic contamination in their drinking water. So especially my research has focused on the arsenic exposure in Chile. So there are many health effects related to arsenic exposure, including cancer and non-cancer diseases, such as cardiovascular disease and respiratory symptoms. But today, I'm going to focus on cancers. So arsenic is classified as a group 1 carcinogen by the International Agency of Research for Cancer with sufficient evidence of bladder, lung, and skin cancer, and some positive associations with other cancers as well. So for my research, I focused on northern Chile, especially the region 2. So here the region is the administrative term, like a state in the US. So they call it like region 1 and region 2 like this. So I focused on the region 2. So this area is very dry. So let's look at this picture. So this area is called the driest habitable place on Earth. And it also has been compared to the surface of Mars. So in Antofagasta, the mining industry kept growing in 1930s, 40s, and many people migrated into this region. But keep in mind, this area is very dry. So later they experienced a shortage of drinking water. So the city decided to add a new source of water from the nearby river. But usually river water is considered as surface water. So we wouldn't worry much about arsenic because arsenic exposure is usually from groundwater. But actually, this water was from the Andes Mountain, and it was geothermally sourced with a very high level of arsenic, up to 1,000 parts per billion. Yeah, so this region in Antofagasta has a very unique exposure scenario, which is an advantage for our research. So since the area is very dry and there were very limited water sources, so almost all of those residents got their water from only one public water system. And Antofagasta is the biggest city in Region 2 of Chile, and around 125,000 residents were exposed to this extremely high level of arsenic. So this is very rare case in environmental epidemiology. Okay, so let's look at the concentration of arsenic in drinking water in Antofagasta. So as I previously mentioned, after they added the new water source in 1958, the arsenic level dramatically increased until they installed the new treatment plant to remove arsenic in 1970. So at this time period, between 1958 and 1970, the average arsenic level in their drinking water was 870 parts per billion. So this level is 87 times higher than current regulatory level 10 parts per billion. So this was a public health disaster. But ironically, this scenario in Chile is very beneficial for us to study the health effects of arsenic, because the exposure level was so high, so we could see the clear association without confounding our results. Okay, so we found arsenic increases the risk of many diseases, including cancers and non-cancer diseases. But while doing these research, I had some questions. So first, I was curious about how long the risk of cancers would last after exposure had stopped. And another one was, I also wondered about the difference by age. So when people were exposed to, especially in utero or during early life. So I tried to fill these gaps. Okay, so I'm going to talk about what I found. So the first one, so how long does the risk last after exposure has stopped? So let's look at this one. So we have very good data for that, a very good record of past exposure in Chile. So in this situation, so they had the distinct period of high exposure between 1958 and 1970. And we had health data until 2010. So we could follow up until recent years, which means 40 years after the exposure had stopped. So let's look at this. So we might have cancer like 10 or 20 or 30 years after exposure. So I'm going to show you what I found in this research. So even look at the most recent year between 2000 and 2010. And this table shows the standardized mortality ratio. So which shows the relative risk of cancer mortality in Antofagasta compared to the rest of Chile. So it shows elevated risk compared to the rest of Chile. So as you can see here, the relative risk of lung cancer, 3.38 times higher. In the case of bladder cancer, 6.43 times. Even kidney cancer, 2.09 times higher risk in this area. So we also observed the historical trend of arsenic-related cancers. So here we compare the rate ratio in region 2 with region 5. So at this time point, why did we compare it to region 5? So mortality data was not available electronically for the years before 1970. So it was not possible for us to collect all of those records in the paper. So instead, we decided to compare region 2 in these years with region 5 that had very low or no arsenic exposure. And it also has very similar socio-demographic demographic factors to region 2. So as you can see here, the blue shaded part shows the period of high exposure, so 1958 to 1970. So you can see the rate of cancers has increased around 10 years after the first exposure. So the risk of cancers increases continuously. So even look at the most recent years between 2000 and 2010. So it still shows very elevated risk compared to the comparison area. So diseases due to the arsenic exposure can have a very long latency with increased risk, like 40 years after the exposure stopped and 50 years after the first exposure. So until now, the asbestos is known to have the longest latency. So for example, if you are exposed to the asbestos today, then you might get mesothelioma after 42 years. But even here, so we could see arsenic also has very long latency up to 40 years right now. So that's why it is very important to keep looking at the risk of disease in this region. In this way, we can know how long the latency of arsenic-related diseases are. So this research was published in a Journal of National Cancer Institute in 2018. So if you are interested in this study or want to read more details, please have a look at this paper online. Okay, so actually during this high exposure time, people were exposed to arsenic at different ages. So I wondered, so what was the effect of exposure in utero or during early life? So let's look at this. So here is the time frame of this study. So again, the high exposure period was from 1958 to 1970. So if somebody was born during this time period, that person was exposed to arsenic in utero or in early life. So people born earlier than 1958 were exposed to high level of arsenic as older children, adolescents, or adults. So we evaluated the mortality from each cancers. But why did we set the study period from 2000 to 2010? Because these years are the most recent data we have now. So another reason is generally in people before age 30, cancer is very rare disease. Okay, so as you can see here, so this graph shows the relative risk of each cancer mortality when people were exposed to arsenic in utero. And the next bar shows the people exposed aged 1 to 10 and 11 to 20 and even up to over 41. So for example, let's look at the bladder cancer. So the risk of bladder cancer is significantly elevated for those exposed at any age. But when they were exposed to this high level of arsenic in utero, the risk of bladder cancer is 16 times higher during their adulthood. So even when exposed at age 1 to 10, almost 8 times higher risk. And in this study, we also found the association between arsenic exposure and laryngeal cancer. So we could observe a 10 to 20% reduction in relative risk for each 10-year increase at exposure. And lung cancer shows a little bit different trend. That's because the lung isn't fully developed until age 20. So that's why, compared to other type of diseases, the risk of lung cancer is still elevated, although they were exposed at the later age until 20 or even 30. So even the kidney cancer, the risk is similar whenever they were exposed. But overall, so we found the arsenic exposure in early life shows a marked increase in cancers. Okay, so you might concern about other confounding factors. So this slide shows that there were no significant differences in demographic, lifestyle, or medical risk factors in Antofagasta compared to other region of Chile. So we also have collaborators in Chile and confirmed that there was no other significant exposure like arsenic in this area. So especially smoking is the major risk factor for many diseases like lung or bladder cancer. So smoking rates were pretty similar between Antofagasta and the rest of Chile. So based on a recent meta-analysis, so bladder cancer specific relative risk by smoking was reported as 1.5. So if everyone in region 2 smokes and everyone in other regions are non-smokers, then the relative risk should be 1.5. But as you could see, so we found that the relative risk of bladder cancer was 16 times higher. So this means our result was not confounded by other factors. And this research was published in the American Journal of Epidemiology last year. Okay, in summary, we can see arsenic-related cancers have a very long latency. So the risk of cancer is still increased 40 years after the high-level exposure had stopped. And we could also find that arsenic exposure in early life showed a marked increase in disease in later life. So based on our findings, we get the message that although arsenic exposure is not continuing, the risk of disease is still elevated. So we still need to focus on this arsenic exposure. We still need to follow up to those exposed before and provide proper intervention, including screening. And we also need to focus on the arsenic exposure in the younger generation. And although I didn't show it today, we could also see that low level of arsenic increases the risk of diseases. So therefore, with more study and evidence, so we should suggest to the government to make the policy more strict by reducing the current regulatory level. Actually, New Jersey and New Hampshire already reduced their regulatory level to 5 parts per billion. So here are my collaborators at UC Berkeley and Catholic University in Chile and Texas A&M School of Public Health. Thanks for your attention.
Thank you so much, Dr. Rowe, for your presentation. We have a lot of questions here, and I just wanted to remind you we'll get to as many as we can. And if we can't, we'll try and pass on the questions to the authors of the studies and see if we can get you answers later on as well. I want to start with a question here. This is to Dr. Shepard. So one of our audience members wanted to ask if the high exposures in people, the subjects studied, were pesticide applicators, or were there other types of exposures?
Well, in all the studies, the exposure groups were, all the people studied were occupationally exposed, so they would have predominantly, yes, been applicators. And in fact, in the agricultural health study, that was the focus, was on registered pesticide applicators. And the case control studies varied a little bit, but they were all occupationally exposed.
Great. And I guess just another follow-up question. Another person asked if would railroad use of glyphosate simulate green burn down in agriculture?
I'm sorry, will you repeat that question?
Yeah, they asked, would railroad use of glyphosate simulate green burn down in agriculture?
Well, green burn down is where it's applied to food crops shortly before harvest. And the reason is to dry it out more quickly so it won't mold in the field. But the problem with that is that the glyphosate takes a little while to break down. And so relatively more of it gets in the food supply when it's sprayed on the crops right before they are harvested, as opposed to the other use in food crops, which is to spray during the growing cycle to keep the weeds down. And so there's quite a bit of time between the spraying and the harvest for the glyphosate to break down. So railroad use would be different because it's not sprayed on food crops.
Great. And this is another one for you, Dr. Shepard. First of all, great presentation. And your meta-analysis was the 41% increased risk for any or all cancer or for just non-Hodgkin's lymphoma?
So we focused exclusively on non-Hodgkin lymphoma. We didn't look at any other cancer endpoints.
Great. And then for Dr. Rowe, I have a question here for you. She said, so one of the questions, oops, sorry, I'm pulling this up now. It would be, did you check to see if there were cancers before age 30 when cancer is more rare?
So in our previous study, we also investigated the effect of arsenic on childhood liver cancer. So compared to the people over age 30, the cases were not much.
Great. Another question for you, Dr. Rowe. Somebody said, I would point out that after 1970, exposure did not stop, but was still around 100 micrograms per liter until maybe 2001. Those levels are still considered high, even though they're lower than the 1958 to 1970 levels. Similarly, the age at first exposure wasn't really that as before 1958. There's still arsenic exposures at around 100 micrograms per liter before 1958. So she was just wondering if it's comparing the high versus the low. Did you consider low dose exposures? And what would be some of the outcomes from low dose exposure compared to the high?
Okay, so in our current view, it looks still very high, 100, because current regulatory level is 10. But even as I mentioned, this cancer risk is relative risk. And in 1970s, at that time, the regulatory level is much higher. So even in the US, the current regulatory level 10 parts per billion was started since 2006. So that's one important thing. And another one is, as I previously mentioned, this area has very unique scenario. because like before this 1958 it was around 100, but during this time period, it was almost up to 1,000. As I mentioned, the average was 870 parts per billion. After that, it dropped down to the same level as before after they installed the treatment. So one thing you can see is based on this distinctive of extremely high level of arsenic, we can draw the curve of relative risk. So based on this one, we could figure out the relative risk of this cancer mortality is still elevated in recent years. So that's one point we need to think about.
Great. Another question for you, Dr. Rowe. What about non-cancer effects of arsenic in this population?
Yeah, so today I didn't show you, but if you read the papers that I talked about today, so you can read about those things. So important thing is we could also find the elevated risk of respiratory diseases such as bronchiectasis or asthma, and even myocardial infarction and chronic kidney failures. So arsenic is related to many different diseases, including cancer and even non-cancer diseases.
Great. And Dr. Prins, I wanted to ask you a question that came in. What would be the endocrine disruptive chemicals other than agricultural chemicals you would prioritize for investigation in the context of occupational exposure studies for prostate cancer? Do you think that prolonged adult exposure, not only perinatal exposures, could increase prostate cancer risk?
Yeah, there is some evidence that that indeed takes place. You said non-agricultural. There is some agricultural data looking at a number of the different pesticides, and then that would only be during adult exposures, showing more that it's like a gene environment interaction going on with a number of the pesticides. So certain genotypes with certain polymorphisms in some genes increased risk of exposures in the adult. And so adult exposure is certainly something that's of a concern. Also arsenic exposure. That's one question I wanted to ask, is if this group in Chile was investigated for arsenic. Arsenic is something we're concerned with, not just early life exposures, but adult exposures as well. We're also concerned about exposures to some of the compounds that are in sunscreens and whether they may be influencing populations of cells within the prostate gland as well. There are a number of other different endocrine disrupting chemicals. Oh geez, there's some evidence for some of the PCBs for dioxin as well, that these might be influencing prostate disease, and it's not just during development. So there is evidence in others. I have some publications, some review articles on this that you could look up in the literature.
Great. And I don't know if all of our audience has gotten to see some of the other questions that you answered initially, Dr. Prins, while the other speakers were finishing up their presentations, but there was one that was a good one, and it just asks if you will explain the rationale for including testosterone in the second hit along with the estrogen, since sea levels.
This is the basic physiology question.
Yeah.
So I get asked that a lot. In fact, if I just give estrogen to the animals at a higher level, that will shut down the hypothalamic pituitary testicular axis. So LH would be suppressed and testosterone levels would be suppressed, and the prostate would just atrophy. And so then you're not going to see, then you're going to see very strange results. So we just give testosterone at physiologic levels to maintain the health of the prostate. And in that background, then we boost the estrogen. So that's a experimental design question based on endocrinology.
Great, thank you so much. And then question for Dr. Shepard. What types of studies do you think should be performed to fill in some of the gaps in existing studies that could shift policy?
would be really hard to do, but I think it would be better, or it would be good to figure out how to address lower level exposures in the general population and not just focus on workers. And then I think that with respect to human studies, that's where I would want to focus. The challenge is exposure assessment. It's extremely difficult to quantify exposure because we're relying predominantly on people's memory. The other thing that I would focus on is higher quality exposure assessment. I didn't talk about the exposure paper review we did, but for the ubiquity of glyphosate, there's a huge paucity of quantifying exposure in, say, urinary markers. And that's in part because the laboratory measurements are extremely difficult to do, and there hasn't been good standardization of them yet. And so we don't really even know how widespread the exposure is in the population. And that would be another important thing to do. And linking that with some epidemiology would be very, very valuable. But I don't think we also know very much about the half-life, and that would be another aspect that would be important to study in the context of these questions.
Great. I guess one other kind of paradigm question for you, Dr. Shepherd, says, could you expand on what a new paradigm to review pesticides may look like?
Well, I'm concerned that when I say that, I'm concerned that particularly with registrant funded studies that there are many different decisions that get made in the design phase and the data analysis phase that could obscure evidence that's there. And so we need to have a more careful assessment of studies that are done for policy reasons, that are used for policy decisions. And I also think that there's a real importance in having independent scientists that don't have a vested interest in the outcome of the studies, conducting a lot of the studies that are done for policy decision making. And right now that mechanism is not in place in the same way it is for agricultural chemicals and that are being considered for, that the registrants have to provide the data themselves.
Great, thank you so much. Thank you to all of our presenters today. I think we're coming to a close with our questions. If there are any that we didn't get to, I'll try and follow up on those later with our speakers. But I wanted to thank our speakers one more time, and also follow up with a few things. The next CHE EDC Strategies Partnership webinar will take place March 24th and is titled Towards a Global Understanding of Chemical Pollution, a first comprehensive analysis of national and regional chemical inventories. To learn more into RSVP, please visit our website at healthandenvironment.org.
April 24, not March 24?
Sorry, April 22nd, excuse me. Thank you for helping me clarify that. And then I also wanted to mention, if you're new to CHE and would like to stay updated about upcoming events and more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate CHE's partnership webinars bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I want to thank our speakers, Dr. Prins, Dr. Shepherd, and Dr. Rowe. Thank you so much for your time in presenting today. Thank you to everybody who joined us as well, and have a great day. Stay safe and healthy.